at this meeting. I'm an applied statistician working at a university. My primary goal is helping my collaborators make sense of their data. Sort of secondarily is to try to develop new statistical methods to better make sense of their data. Sort of thirdly, to implement those methods in software. For nearly 20 years, I've been working on an R package called qtl, and I want to tell you about that experience. So that the idea for this package came from a friend of mine visited, and we were talking about the need for sort of specialized software in sort of the applications that we worked in. It was actually the week that R version 1 came out in January of 2000. He wanted to write it in MATLAB, and I wanted to write it as an R package. And I was 5 years younger and devoted more time to it, so it became an R package. This figure shows lines of code over time. Each dot is one CRAN release. So it was first on CRAN at the end of 2001. It's grown to be nearly 40,000 lines of R code and 25,000 lines of C code, and over 15,000 lines of the .rd documentation, which back in the day we typed by hand. This didn't actually, for the first 7 years, it was without version control. The way that I kept track of versions is by tarring up the file at its current state and just saving that, tar gz file. I didn't get into version control until 2008. So it was somewhat, so you'll see what my experience has been, I guess. But I feel like I should tell you a little bit about the package, and so in doing so, I really need to tell you a little bit about the science. So I study genetics of complex traits, mostly in mice. Mice have a big advantage for genetics in that you can make inbred lines. So you can take a pair of mice and mate them, and then do repeated mating between siblings for 20 generations or 200 generations. So what you get at the end is a strain of mice that are all genetically identical, and that if you can figure out which one's a male and which one's a female, cross them, the offspring will all be genetically identical again. The advantage for genetics really is simplifying a lot of things and being able to make really large crosses. Our interest in the genetics in mice is really focused on understanding the genetics of human disease. We're not trying to cure disease in mice. We're trying to have a better understanding of the underlying biology of human disease. The basic experiment that's done, it would be an intercross where you take two strains of mice that differ in some trait. Like one strain has high blood pressure, the other strain has low blood pressure. You're trying to find what genes are contributing to that difference in blood pressure. So you do a cross that's identical to what Mendel did with his peas to mix up the high blood pressure chromosomes and the low blood pressure chromosomes. And then what you're going to do is measure, get a bunch of offspring, measure blood pressure in all those mice, which really for that phenotype would be the hardest part of this study. Measure the blood pressure of all those offspring and measure genotype along each chromosome, whether a mouse is blue-blue, pink-pink, or blue-pink at a given position. And then we're going to scan across the genome and look for regions of the genome where there's an association between what genotype you have, whether you were blue-blue, blue-pink, or pink-pink, whether that's associated with the outcome blood pressure. So the results of this sort of analysis is a plot across the mouse genome, on each chromosome, the curves that for a test statistic that's indicating when this thing is high, you are saying there's evidence for an association between genotype at that location and blood pressure. Those peaks we call qtl, or quantitative trait loci, a region of the genome that is affecting a quantitative trait, blood pressure. But effectively what you do is at every position in the genome, you do an analysis of variance to see is there a difference in the average trait across the 3 genotypes. So, and that's, the goal of my package, RQTL, is called QTL because we call these sites we're looking for QTL, or the genetic loci that affect this quantitative trait. It does a bunch of other things. But yeah, going back to the package, and I just want to talk a little bit about, the state of it and my experience with it. 20 years into a project, there may be some good things about it, but they start to become harder to identify. Some of the code in this package is really good, and I'm proud of it. Some aspects of the interface, the user interface, the code that the user types to get something to happen, I think was a good idea that I had back in 2001. It includes a lot of data diagnostics features and data visualization. And it's quite comprehensive. I had this idea that I would implement all methods, good and bad, so that you would have sort of one place that you could try things out and understand them. And it's quite flexible that, I can implement new ideas about, for analyzing data with this. Other people could potentially do so as well. But it's easier for me to think about the bad things. The first example I want to give is just about data, getting data into R for this kind of cross. So these sort of data, I need, for this sort of experiment, I need a set of traits. I need a set and then genotype data of are you blue, 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 pink, pink, pink at sites along the genome. And then I need to know where those positions are in the genome. And the main input format we use is just a comma delimited format where the first few columns are all phenotypes and the traits, and then the remaining columns are all positions in the genome and their genotypes. And the first row has, for those later columns with the markers, the sites in the genome, it has their chromosome ID, and the third row has their position. And the first thing I do reading this in is I need to split the trait data from the genotype data, which I do by looking for, where's the first non-blank cell in that. It sort of, as RQTL was used, people started to load in data with lots of traits, like say 25,000 traits. And they would ask me, why is it taking so long to load the data? And I would say, how much data do you have? And they'd say 25,000 phenotypes. And I'd say, it's going to take a while. It's a big data set. But then I had a collaboration where I had data that was, 20,000 traits, and it took, it was like 20,000 phenotypes, and then the usual kind of markers, and maybe 300 mice, and it took a minute to load this CSV file into R. And I was like, something's wrong. It shouldn't take a minute to load a file that's really not very big. So I looked into the code, which I declare to be the stupidest code ever. This code is trying to find the first non-blank cell in that sort of second row. It does that by looping from 1 to n, where n is the number of columns. And it first says, is the first cell blank? Is the second cell blank? Are the first 2 cells blank? Are all 3 of the first cells blank? Are the first 4 cells blank? Are the first 5 cells all blank? When that fails, then it breaks out of that loop. And then it does a bit of extra work of like trying to find that, so in loading my data with 25,000 columns of traits, that took about a minute. It was about 58 seconds was spent on this for loop, and 2 seconds was actually the loading the data and everything else. So I like to say, open source means everyone can see my stupid mistakes. And version control means everyone can see every stupid mistake I've ever made. But, so that's not the only bad. That code is no longer in the package. But you can find it in version control. It's on GitHub. But there is other bad code that's in the package. One of the R functions that does sort of a major thing of a two-dimensional scan of the genome to try to look for a pair of these qtl is 1,400 lines long, which is not, I mean, the function is 1,400 lines long. In the C code, I mentioned that this package has almost 25,000 lines of C code. Almost 20% of that is related to this one sort of thing that I'm trying to do. I mean, it's complicated, but it's also complicated and not really, you know, I mean, yeah. It's not what I would do today. The package, I'm really happy with the interface for the users. I'm less happy with maybe the data structures that I set up in the year 2000. There's some really pretty Baroque data structures, to put it nicely. This is a bit of code that I actually do type because it's something that I want to do. So there's some stuff that you want that's kind of deep inside some really complicated object. There are lots of inconsistencies in the code, and like you have functions that are doing kind of similar things that have names that don't, like, they're not designed. They're not designed. And they have arguments that are really the same argument, but that the two different functions have different names for that argument. You know, a marker might be marker, or it might be mname1. I would say that maybe the biggest mistake I've made, it has been really useless warning messages. A lot of users of RQTL, this is their first experience with R. Their first experience with software other than Excel and web browsers, maybe, and Word. So they get a warning message, x prime x is singular. I mean, what are they supposed to do with that? What does that mean to them? And so a lot of the questions I get are, I get this warning message, x prime x is singular, what should I do? And I think, I was looking at this yesterday, I think x prime x, you know, I know what that means, but that could be a picture of something, really. To someone that doesn't know the details of linear regression, x prime x is singular. I mean, parsing that sentence is difficult. Tests. Tests. There's not very many tests. I actually said this. I don't need tests. I have users. Janny Bryan has said, if you use software that lacks automated tests, you are the tests. And it brings me some comfort that we agree. You know, Hadley, his paper about unit testing and the testthat package, it's not that we don't test our code, it's that we don't store our tests so they can be rerun automatically. And this, to me, is just like the essence of Hadley. I mean, this is exactly why I love Hadley. Because he starts it off by saying, you know, it's not that we don't test our code. So it's not that we're like terrible people. It's just we have room for improvement. So, yeah. But how did this happen? How did I spend 20 years trying to, you know, develop and maintain and support a package that has these problems? You know, I didn't know any better. I didn't know any better at the time. I mean, I started to know better, but by then it was like too late, it seemed like. There was not enough rewriting done because, I mean, I have the to-do list. I have a to-do list in here that's just absurd. I just like none of that stuff I'm ever going to do. So going back and redoing stuff that I've already done just seems like, I mean, it works. I don't have any tests that would prove it, but I did test it. There's not enough planning. I would say, you know, I mean, personal weaknesses. There are trade-offs to make that maybe I made the wrong choices. A couple other quickly things that I've learned making RQTL is that when you're writing documentation for a package, what users care about is tailored examples. Show me your package in action doing exactly what I want to do, so I could like plug in my data at the top and run it, do just what you did. And so if you're going to put effort into documentation, it's really think about how people are going to use it and focus on that first. Kind of a general user guide, sort of second, examples within the detailed documentation third, formal documentation, it seems like it's important, but no one, seems to read it. User support, I put a lot of effort into answering people's questions. I get emails a couple times a week at least still. I think if you want people to continue to use your software, you really need to help them to get started. And so, I really want to make myself useful. If they need help getting their data onto RQTL, I'm happy to have them send me the data and I'll figure out what's going on and send them code and their data back, or fixed code. Fixed data that works. Going back to QTL mapping, sort of the main target is to try to find a gene, to sort of isolate a position in the chromosome where the gene that's affecting this trait is sitting. And those regions are too big, so sort of modern experiments of this kind are really focused on trying to break down the chromosome smaller by doing more generations and sort of mix together more than two strains. And neither of these really works, can be studied effectively with the current package. And so, and also the size of data sets have increased enormously. So I've been focusing on RQTL2. Now as a Minecraft plugin that basically starting over completely to try to be able to handle high dimensional data, to be able to handle these multiple strain modern crosses. And I'm trying to avoid making the mistakes that I made the first time around. Rcpp, Roxygen 2 unit tests with testthat have been, just make everything work better. My life is so much nicer now. But I still have some personal failings, so RQTL2 is not like, not without its flaws. Finally, I've devoted a lot of my time to software, even though sort of the people that evaluate my work don't really care about it at all, because they just care about grant money and citations, papers. But it has actually been really helpful for my own scientific work to find new collaborators, and it just, statisticians, our key job should be to make ourselves useful, and this has really been sort of the way in which I've been most useful, I think. A lot of people involved in it, and I'm a bit.ly squatter if you want to see these slides. They're at RStudio 2019 at Bitly. Thanks very much.